In this video presentation, I will cover the paperwork required for your science fair project. Every student grades 6 through 12 submitting a project to our science fair must submit Isef forms. This is especially important in grades 6 through 12 as the projects are available for other competitions such as the Thermo Fisher Junior Innovators Challenge, the Junior Science and Humanities Symposium for grades 9 through 12, and the Regeneron Science and Engineering Fair. The goal of the video is to point out some oversights when completing the Isef paperwork and to address some problem areas that many students experience. When starting Isef paperwork, it is important to understand that all regional fairs following the international rules and guidelines set by the Society for Science must be followed. We are required to adhere to these rules to maintain our affiliation through the Society. I will review every Isef form available to students. To begin, it is important to know that all forms except Forms 1C, 5B, and 7 and the abstract are completed after the research is finished. Additionally, dates of the signatures reflect when approval or consent was given. Please use the format set by Society for Science as the month, the day, and then the year format for all your dates. The checklist is provided so that the adult sponsor can review what information and therefore which forms must be provided. Student or student names must be written here, so that includes teams. Project title needs to be here, complete. This box, number 1, number 2, and number 3 will be checked by everybody. Only projects that are human, vertebrate, PHBAs, or microbes would check number 4. And then all student projects will check every box of the adult sponsor, the checklist 1A, the research plan, and the approval form. If your project involves humans, therefore number 4 would be checked, you would go down and you would check humans and then what you have included with that particular project. If it's vertebrates, you do the same thing and check the boxes, PHBAs, so on, and then hazardous materials that they might use. Other would be that risk assessment. Every student checks down here to attest that they have checked the above boxes in red and abide by the rule. The other part that you want to make sure is the date. Make sure that the date is in the format. The sponsor has their name, their signature, their phone, and their email. If an electronic signature is done here by Adobe, it will timestamp it. If it timestamps a certain date that is after the project has begun, the project could risk being disqualified. On Form 1A, Student Checklist and Research Plan, the student outlines what the project is about. Items that especially need to be clear are the following. You need to make sure that you're clear about the schools, and if there's schools that are students that are working from different schools, all those schools need to be listed. If it's a continuation project, you need to make sure that you check yes, and that you need an abstract and your research plan from your previous year, and you have to complete the continuation form 7, which we will cover later. Make sure that you're only recording and giving information from the current year's research, because that's all it's allowed to be presented. That's why you have to present the last year's abstract and research plan. The current year's project must demonstrate significant progress to be considered a continuation. And number 7, the dates when you're looking at this, make sure that you're looking at the date that you started the actual project and not the background literature or any other aspect of the project when you started your experimentation. And it can only be a 12-month period that occurs before Isef is allowed to accept that project. For this one, where did you conduct it? If it was at a research institution, a school, the field, the home, universities, research facilities, and industrial settings will require different documentation, which is Form 1C. And so we will cover that in just a little bit. Pathogens, any kind of microbes cannot be cultured at home, so you cannot, make sure that you know that you can't do that. Make sure that you write down any non-home or non-work sites, give all the information, an email address, and the phone number. The last part you want to look at is the research plan and the summary, and we will cover that in just a minute. Make sure that you cover all that information in that plan, so that you'll cover all the details that is required by Isef rules. So an overall summary, this is done by all students. You need a tentative start date. You need an actual start date that doesn't include just background research, but when you actually start the experimentation. You need an end date. You can work on a project after regionals to state, but only to add to the existing data. You cannot add new variables or any other aspect. You cannot change the project in any way. You can only add to your database. The research plan summary is a very important aspect of the science fair project. Isef requires every student, grades 6 through 12, to submit a research plan with these components. These components include your rationale, why you're going to do it, your research question or questions, your hypothesis or hypotheses, your engineering goal or goals, if you have more than one, and your expected outcomes. You also need a comprehensive list of materials that you will be using. Make sure that your procedures are thorough and that any risk or safety issues concerning the project should also be described. You also need in there your bibliography or your works cited page, and make sure that they're not links, but it's actually a citation using APA or MLA format, which we will discuss in just a minute. If a student is actually conducting a human subjects, these projects here, then they would need to include all this information on their form, and we'll talk about that as well. Same thing applies for vertebrate animal studies. You would cover parts A through F, and if there's potentially hazardous biological agents, that needs to be included as well. Any hazardous materials, activities, you're going to use a bow and arrow, you're going to use a chainsaw, you're going to use a drill, you're going to use a hammer. All that stuff needs to come under risk assessment and needs to be, a form needs to accompany that. This research plan template can be used to write your research plan and is available on the State Science Fair website. Each text box will expand to accommodate your explanation. So each one of these will drop down and you can expand and add more information to that. The first area that I have circled here is required by all students. Details are important. If a project deviates from the research plan while you're working on it, then you need to update the plan and resubmit. The subject-specific items such as human participant details what is needed in the text box. So everything here, we need to have all of this information in this text box. And again, it will drop down and allow you to add. Make sure that all your materials, your survey, anything that you're going to use is included in that. If your project does not involve humans, all you're going to do is highlight it and remove it from the document. For projects involving vertebrate animals research, PHBA, which is your microbes, and your hazardous material activities or devices, you're going to fill out these boxes. So vertebrate animals, PHBAs, or your hazardous materials, you need to put that information in those boxes. The procedure, if you do not need to use the vertebrate animal or the PHBA, I would suggest that, again, you highlight and remove. Most students have hazardous materials or some sort of device that they're going to use, so it won't all the time be removed, but it can be removed too as well if there's nothing in your project that required it. The procedure, data analysis, and conclusion, and the site of the experimentation, both of these areas need to be included, and that's for all students. Be sure to include the information about how you're going to clean up your research, meaning if you've got microbes, you're going to use the aseptic method for cleaning up biological contaminants and where you're going to get rid of them, and disposal should be included. If it's a continuation project, then you're going to fill out this part on your research plan. If not, then you're going to highlight and you're going to get rid of this as well. The last part is your bibliography and your works cited page. This section is required by all students as well. You may make sure that all your references reflect your project's topic. For example, Isef wants to know that you researched a biological agent that you actually used in a microbiology project. And again, you'll include that in your drop-down box, and it will continue to grow as many resources that you have. If you have vertebrate animals, human subjects, or PHBAs, you have to make sure, and chemicals, you've got to make sure that you've got a citation for those aspects of your project. When writing your bibliography or your works cited information, no links to websites are acceptable. So that means that the citations need to be cited either using an APA format or an MLA format or other citation types such as Chicago or Turaban writing style. The information on how to write the bibliography works cited can be found at the Al Purdue website, or you can use the American Psychological Association website, and both links of those are at the bottom of this page. So on Form 1B, this is your approval form. This is including all team members. These statements are going to attest that each of the people in the committee or those consent to the project. The dates should also be signed as described below and are before experimentation, otherwise indicated. So when we look at it, it's got to be signed before. Student date means they attest to and they understand the possible risk, that they have read and they will follow the rules and that they will abide by the ethics statement. And the parent and guardian date means that they contest that their child is doing a project. The next aspect is your SRC approval, and that comes before the committee reviews the project and before the experimentation. So making sure that everything, when we look at this before that date, we've got to make sure that the dates coincide with before that project actually gets started. Projects must be pre-approved that are in the areas of human subjects, vertebrate animals, and PHBAs. By those designated scientists, including microorganisms, recombinant DNA, and human and animal tissues. SRC approval after, when we look at it, so this is our before and our date that's prior to, and SRC after, when we're looking at it, are projects that needed pre-approval by the SRC but were done at a research institution. So if you're not at a university, you don't even need to look at that box instead of the affiliated science fair person. The date signed indicates when the SRC approved this project after it was completed. All documentation from the research institution showing approval of the project must be attached. So there could be forms that follow that. The final SRC projects must be reviewed by the SRC after completion, after the experiment is complete, and shortly before the competition of the fair. So again, this down here has no bearing on you. This is strictly done by your regional fair director and their SRC committee. This slide is to help clarify the difference between a direct and a qualified scientist. A qualified scientist has earned a doctoral or professional degree in a specific discipline and or has expertise and extensive experience in the student's area of research. The direct supervisor does not have an advanced degree, but must be familiar with the student's project and agree to the training. The qualified scientist can also be the adult sponsor, as well as the direct supervisor can also be the adult sponsor. The qualified scientist is required to complete the Form 1C, which is the regulated research form, the qualified scientist Form 2, the risk assessment Form 3, if it applies. They also oversee training the student at the research facility and or direct supervisor. The direct supervisor provides supervision of the student's experimentation and completes the direct supervisor box on Form 2 when it's applicable. They must also review and complete the risk assessment form when needed. So this form is completed by the supervising adult, the qualified scientist, and explains what the student researcher actually did and is signed after the project is completed. The form is only needed if the research was conducted at a research institution, a university lab, for example, or in an appropriate industrial setting. It is not completed for work that's done at home or at high school. Remember to record the date as month, day, and year. So make sure that you have your direct supervisor's name, make sure that you have the date, and make sure that all of your boxes are checked. Don't leave anything unchecked. On this page, the Qualified Scientist Form 2 explains what will be done to oversee the project. The qualified scientist and, if needed, the designated supervisor will sign the date that they approve before experimentation. Make sure the dates of approval are prior to the experimentation, and that every box is checked on the form. So this includes when they go through, you can't leave any of these blank. And again, any electronic signatures will be time-stamped, so make sure that your dates are prior to the start of the experiment. Form 3, the risk assessment form, is required for projects using hazardous materials, activities, and devices, and is also done before. Some PHBAs, including protists, composting, coliform kits, decomposition of vertebrate organisms, and microbial fuel cells also must be completed and signed by the designated supervisor or qualified scientist before the experimentation. In a statement from Isef, in the U.S., the Food and Drug Administration, the FDA, tightly regulates the issuance of prescription drugs, including non-controlled medications. State law further regulates the use of prescription drugs, and it is unlawful for any person knowingly or intentionally possessing a non-controlled medication unless it was obtained directly from a valid prescription or order of a practitioner while acting in the course of the professional practice. It is also unlawful to use a prescription for persons or purposes outside the original intent of the prescription or for the person it was originally prescribed for. All applicable federal, state, and country laws must be followed. Exceptions include research and educational products purchased that are considered research grade and not pharmaceutical grade, therefore not for human consumption. Prescription drugs administered to vertebrate animals may only be done under a veterinarian's supervision and with prescriptions provided for this specified purpose. So as you're looking for it, make sure that you've got anything that's hazardous materials. Make sure that your dates also coincide. Form 4, the human participant form, along with the research plan, is submitted by the student researcher before the experiment to explain to the IRB how the safety and well-being of the test subjects will be conducted and the confidentiality of the results will be ensured. The IRB reviews the project, checks the risk level, and determines if the written documentation of assent, consent, and permission is required. All questions must be answered and boxes checked and each IRB member signs the date they approve the project. When required, the IRB, a written informed consent parental permission form, is used to explain the research participant and their parent or guardian the risk and benefits associated with participation. A human informed consent form template is on the next slide. Questionnaires, sample tests, etc. must be given to the IRB and to the parent guardian. If the participant wishes to participate and when required the parent also agrees, they each must sign the informed consent with the date that they approve. Again, this begins at the beginning of the experimentation. Students are prohibited from providing advice, diagnostic, or medical information to participants without direct supervision and involvement of the medical professional. This includes publishing diagnostic apps on public websites and app stores without appropriate FDA approval. So make sure that you've got all your boxes checked. Make sure that the IRB has completely filled out every aspect of the form and that you have all the appropriate signatures. So the Human Informed Consent Form template is a template that students can download and use, or they may create their own. However, they must make sure that they have the appropriate statements that are on the template on their own created form. No statement can be omitted. Make sure that signatures are obtained at the time of the experiment as well. So you really want to be very thorough in what the purpose is. You want to make sure that as you're looking at this purpose, you're telling them exactly what you do, what they're going to be asked to do, how long it's going to take. You're going to put any kind of risk. Even if there's no risk, you need to state that there is no risk. Make sure that you list the benefits, how they're going to keep you're going to keep their information confidential, and then make sure that you have your adult sponsor and their phone number and email posted on the form. Form 5A, the vertebrate animal form, is when students are doing something at school, at home, or on a field site. It is to be filled out by the student researcher when the experiment is not at a non-regulated site, and they're going to care for those animals. The SRC reviews this document and determines the level of supervision required for the study and signs and dates it before the experiment begins. The bottom of the form is filled out by the veterinarian and/or designated supervisor and is signed and dated when they approve this project with the housing conditions before experiment begins. If there is any illness, unexpected weight loss, or death of an animal during experimentation, the cause must be investigated and a letter from the qualified scientist, designated supervisor, or a veterinarian which documents the situation and the results of the investigation must be attached. Again, the dates reported should follow the same format as all the other formats. So make sure it's done beforehand. Make sure that we have our appropriate signatures by our SRC committee. And again, that's what this part here is stating. Make sure that we have the veterinarian and if we need our direct supervisor. Form 5B, the vertebrate animal form, is only if you are in a regulated research institution, and that is filled out after the research is conducted at the facility. A copy of the IACUC approval, not a letter from the qualified scientist or principal investigator, must be attached. So you have to have the actual approval form. If there was any weight loss or death of the animal during the experimentation, the cause must be investigated and a letter from the qualified scientist, designated supervisor, or veterinarian that documents the situation and the results of the investigation must be attached. Follow all the same date formats as the prior forms have done. So we're doing this afterwards, and we're looking at it as the qualified scientist, and we're writing in our dates from our principal investigator. This form 6A, the PHBA risk assessment form, is filled out by the student researcher and is required by all research involving microorganisms, recombinant DNA, and fresh and frozen tissues, including primary cell lines, human and other primates, established cell lines, and tissue cultures, blood, blood products, and body fluids. The SRC, the IACUC, the IBC, the RAC approval before experimentation. The qualified scientist will sign and date. The SRC will choose one or more of the statements that describe the approval process for the study and will add the date that approval occurred. All local, state, and national laws and permits requirements must be followed regarding the transport and use of microorganisms, such as, but not limited to, citrus greening and tobacco mosaic virus. All studies involving human and wild animal blood and blood products, except those that only involve blood from student researchers, should be at a minimum of a BSL-2, done under the supervision of a qualified scientist. That means not at home. Exempt studies that require no SRC pre-approval required are studies such as E. coli K12, which is the OP50 food used to feed C. elegans. However, include the strain, the source, the quantity, and the biosafety level risk of each microorganism. Date format is going to be the same, and Section 4 is only utilized by the regional and state fair, SRC. So make sure that this is done before. Make sure that when you're looking at your Section 3 that one of these is documented or and given approval for. Make sure that your dates coincide with before experimentation, and make sure you select a BSL-1 or a BSL-2. So Form 6B, Human and Vertebrate Animal Tissue Form, is filled out by the student researcher and explains the source of the tissue. The qualified scientist or designated supervisor signs and dates to document the source and handling of the tissue. And again, this happens before experimentation. If human tissues are used, attach a copy of the IRB approval and fill out Form 6A as well. Under the statement that I have underlined here, de-identified means removing information from the data set that can directly or indirectly be used to determine the identification of someone. Use numbers, letters, symbols, whatever you want, but don't identify the source of your tissues. So make sure that all of these, if you have to do this, you have to do 6A as well. Make sure that you're doing it prior to experimentation, and make sure that you've either checked the first box or the second box. The last ISEF form is when you're working on a current project in a similar area of research, and as any previous project of the student or the team member, it is considered a continuation. Explain as completely as possible how the project is different from previous experimentation, because only the new and different research project is allowed. The date signed is the date the student research is certifying that the information is correct. The previous year's abstract and research plan summary information must be on the form, and using the additional form for any previous year or earlier information. So make sure that you have the previous year's abstract and the research plan that was accepted by your fair, and make sure that you sign certifying that all this information is correct. This last slide gives a detailed list of all of the regional science fair directors for the state of Mississippi. There are 7 fair directors, And if you have any questions about any of the forms, feel free to reach out to your regional fair director to answer your questions. Thank you, and I hope that this was helpful.